Hello and welcome back to PBH435. Today we're going to be covering our lecture on Chapter 4, the nervous system. We've got some important concepts to cover here today, starting with the concept of homeostasis. We'll talk about how this affects the way that drugs interact with various bodily systems. We'll talk about some important brain structures, the glia and the neurons, and also we'll walk through the action potential, the way that neurons communicate with each other. We're going to talk about the branches of the nervous system and the neurotransmitters that are associated with each of those. And then we're going to talk about some important associations among neurotransmitters and how they connect with those brain structures, certain chemical pathways, and the major functions of those neurotransmitters. Finally, we're going to talk about the life cycle of a neurotransmitter molecule, and then we'll wrap up with some important concepts related to brain imaging. So it's important that we understand the functions of the nervous system, and the reason that we go over them here in our class on health studies of dangerous drugs is because basically this is how drugs interact with our body. They affect our nervous system. Our bodies in general like to maintain a condition called homeostasis. This may be something you've encountered in other kinds of classes, but for those of you who hadn't, basically our bodies like things to remain the same, whether we're talking about temperature, the water content of our body, glucose concentrations, acidity. Our body has a certain standard that it likes to maintain. A lot of us are familiar with the example of body temperature being 98.6. If we start to get too hot, our body starts to sweat to cool off. If we start to get too cold, our body starts to shiver to warm up. So our body likes to maintain that internal standard within certain limits. Now, when we start talking about the use of drugs, many of them are going to influence that status of homeostasis in some way. A great example of this is one that some of us may be familiar with. It involves consuming alcohol and the need to urinate. So alcohol inhibits the release of vasopressin. Vasopressin is an antidiuretic hormone. So after alcohol is consumed, more urine is produced. And we see actually some interesting effects on homeostasis depending on how accustomed a person is to drinking. To someone who doesn't drink very often, compared to someone who doesn't drink very often, a heavy drinker is going to produce less urine because of that effect on vasopressin. Now, during alcohol withdrawal, heavy drinkers will again see that difference response. They'll see increased vasopressin release. So that relationship is about homeostasis. It's about the body trying to maintain a certain normality in terms of its water level. We're going to be talking about a couple of different components of the nervous system today. So we're going to be digging into some details. Again, for those of you who've been through biology classes or classes that might be related in other topics, some of this may be review, but it's important that we all understand it because of most of the drugs that we study are going to be affecting the nervous system in one way or another. So when we look at the nervous system, we do have two major types of cells that we see. We see neurons and we see glia or glial cells. We'll talk a little bit more about both of those. So talking a little bit more about neurons. Neurons are those primary elements of the nervous system that are going to analyze and transmit information. We'll talk a lot about how that actually happens. But basically, before we begin, we're going to break those neurons down into their individual parts. And for neurons, we've got 4 defined regions that are important: the cell body, the dendrites, the axon, and the axon terminal. So the cell body, pretty self-explanatory. We'll see an image in just a moment. Dendrites. Now, the role of the dendrites is to receive information or to receive signals. And then the axon, which does the exporting of the signal, and then the axon terminals. So we'll look a little bit more about those in a moment. But there are actually more than 100 billion of these in the human nervous system. So for each of us, these 4 major regions occur over 100 billion times in each of our nervous systems. And again, if you're looking for a way to remember, dendrites receive and the axon exports. Let's look a little bit more into how that works. So here's the schematic of the neuron with those major parts we talked about. We've got the cell body right here. We've got the axon. It's inside that sheath right there. Axon terminals down at the end. And then the dendrites back here towards the left. So in each of these, for each of these four major regions, you can see how they fit together in this schematic of the neuron. Our other major type of cell are the glial cells. This is actually from the Greek word for glue. Glial cells are found around neurons, and they provide them with support and protections. Actually, 90% of the cells in the human brain are glial cells. They form myelin. Myelin increases the information processing speed and efficiency of neurons. Have you ever heard about the chronic disease of multiple sclerosis? What we see in multiple sclerosis is an issue with the production and sustenance of myelin. So we see deterioration of the myelin sheath over time, and people who are experiencing advanced MS, they have difficulties with that information processing, both its speed and efficiency, and they can end up with motor control problems because of that. So until recently, we thought that the only role of glial cells was that structure and support, and we've actually recently learned that glial cells are actually able to communicate with other glia and with neurons. So this is definitely an emerging area in science. Another really important role of glial cells is to create the blood-brain barrier. We're going to talk a lot about that here in class. Basically, its function is to protect the brain from toxic chemicals. We want to make sure in many ways that our brain is protected from the things that enter our body, and that's what that blood-brain barrier will do. In order for a drug to actually have an effect on our brain, to become psychoactive, then those molecules have to be able to pass through the barrier. And we'll talk more about that and exactly how that would occur. So neurotransmission, this is that process that information gets passed from one neuron to the next. This is one aspect of that nervous system function that is really important to understand the effects of drugs on the body. So we're going to talk about an event called an action potential. And the best way to describe this is that electrical signal that gets transmitted across the axon. If you think about a neuron at rest, there are going to be more potassium and negatively charged anions inside the cell and more sodium and chloride ions outside the cell. So the ion channels are going to open and close. An action potential is generated. The neuron is depolarized, and then the message is passed along. So we've got that resting potential where there's that uneven distribution of the ions. And then the ion channels open, and we see electrically charged particles moving inside the cell. And then the neurotransmitters are released. So I've got a quick video that I want to show you that actually animates this, might be helpful in understanding that action potential event.
That is not transmitting nerve signals. What happens when the cell is stimulated? A stimulus changes the permeability of the cell membrane to sodium and potassium ions. This alters the distribution of charge in the cell body, and if the change is great enough, it triggers a signal, called an action potential, that moves down the axon. How does the action potential move along the axon? Voltage-gated channels in the membrane open and close depending on voltage changes across the membrane. When no nerve signals are being transmitted, these channels are closed. A stimulus causes voltage-gated sodium channels to open, and sodium ions rush into the cell. The cell becomes positive on the inside and negative on the outside. Very quickly, the sodium channels close while voltage-gated potassium channels open, allowing potassium ions to rapidly diffuse out. The cell returns to being positive on the outside and negative on the inside, and the potassium channels close. Meanwhile, the sodium ions inside the cell have diffused to adjacent areas, causing a slight change in the polarity of the membrane ahead of the action potential. This change in polarity causes the voltage-gated sodium channels along this part of the membrane to open. Again, sodium ions rush in and the action potential spreads to the adjacent part of the neuron. In this way, the action potential travels down the neuron like a wave. In the wake of the action potential, potassium leaves the cell, restoring the negative charge inside the neuron. Meanwhile, the sodium-potassium pump has been shuttling sodium ions out and potassium ions in, re-establishing the resting potential distribution of sodium and potassium ions. Let's look at another action potential as it moves along the membrane. This time we will measure the voltage changes that occur in one spot along the membrane. A slight change in polarity causes the voltage-gated sodium channels to open. Sodium ions rush in, causing a reversal in membrane polarity. Sodium channels close and potassium channels open. As potassium ions rush out, the membrane returns to being negative on the inside. The sodium-potassium pump then restores the balance of ions present at resting potential.
Okay, so if you've ever heard about an electrical signal moving through your nervous system, that's how that actually works. And remember, you have more than 100 billion of these neurons in your system, and they're constantly doing that all day long. Whether you're seeing or breathing or scratching your nose, all of those events are involved millions of electrical signals moving through your system, so millions of those action potentials. We'll continue to talk about those throughout the semester because, again, this is how we see the nervous system disrupted by drugs.